great time to now shine a spotlight on one of the biggest health issues out there, cardiovascular disease.
According to statistics in Taiwan, many older adults are affected by CVDs. The number of confirmed CVD cases is very high.
Heart-related problems, many of which could actually be prevented. That's why experts are reminding us to take better care of our hearts.
Hi everyone, we're the Heartbeaters and we'll be introducing you to our DNA-based biosensor for detecting cardiovascular disease-related microRNAs.
Let's start with a quick reality check. What if I told you that feeling fine isn't the same as being healthy? Picture this. Lucas, age 67, just retired. He eats fairly well and he still jogs every now and then. He seems healthy. However, one morning at breakfast, he suddenly feels chest pain. Within minutes, Lucas collapses. This seems dramatic, but this scenario isn't rare at all. Cardiovascular diseases is the #1 cause of deaths worldwide, and it claims almost 20 million lives every single year, often without any warning. Now here's the bigger picture. As our world enters aging and super-aged societies, the risk only multiplies. It's not just Lucas. It could be our parents, our grandparents, our loved ones. But here's the hopeful part. Much of this can be prevented. While existing methods of detection are beneficial and effective in some ways, they fall short in others, such as detailed imaging tests being costly or protein blood tests being unable to show heart structure and blockages. That's why it's important for us to identify a potential and alternative biomarker to help us ensure accuracy and address existing gaps in early detection. That's where the Heartbeaters step in. In the 2024 Nobel Prize in Physiology and Medicine, scientists emphasize the importance of microRNA in regulating gene expression. These small, non-coding RNAs are synthesized at an early stage in the central dogma, meaning there are less factors that could affect its production. Therefore, we're presenting you with a detection kit that targets microRNAs as an alternative biomarker, allowing for early detection of cardiovascular diseases in an affordable, accurate, and accessible way, challenging existing problems in the healthcare system. Now, let us take you through how our design actually works. Now I'll be introducing the detection workflow of our product. As mentioned in our introduction, our biosensor will help indicate to users the risks of contracting cardiovascular diseases. But what exactly is a biosensor? A biosensor is an analytical device that detects changes in biological processes and converts them into signals. How this works is that when a target analyte is present in a sample, a recognition element will detect its presence, transducing further processes that result in a readable output shown on our electrochemical reader. We have designed a specialized three-way junction system as our recognition element for microRNA, followed by specialized G4 hemin complexes that transduce the production of electrical signals that can be converted into a readable output. Now, introducing the first stage of our assay, this is the three-way junction binary probe system. The three-way junction is essentially a DNA probe that recognizes and binds to target microRNA analytes. When the cardiovascular disease-related microRNA is present, the microRNA will hybridize to both the B strand and A strand, triggering a strand displacement reaction that displaces the linker from the three-way junction. Based on our design, the three-way junction is formed with the linker displaced only when the microRNA is present. When the microRNA is absent, no three-way junction is formed as the linker remains hybridized to the original B strand. So where does this displaced linker go? The displaced linker turns on our next stage, the split G4 switch system. The G4, or G-quadruplex, is a guanine-rich DNA structure with a distinctive secondary structure. In our assay design, the G4 is split into two separate DNA strands that can't function on their own. When the displaced linker is present, it will hybridize to the split G4 strands, increasing the overall stability of the split G4 complex. Due to its enhanced stability, hemin will bind more strongly to the split G4. In contrast, when the displaced linker is not present, hemin's hybridization is comparatively weak. The split G4 hemin's interactions with TMB and hydrogen peroxide catalyzes a redox reaction that produces electrical signals. These electrical signals will be converted into a readable output using an electrochemical reader that will indicate to users the risks of contracting a cardiovascular disease. After the redox reaction of G4 hemin complex and TMB occurs, electrons will then be released, in which electrical chemical signals will then be made. These signals will then be detected using cyclic voltammetry, and electrical chemical methods that sweep voltages across the electrode and measure current changes in real time. A sharp oxidation peak will only be observed if targeted microRNA triggers a strong electron loss. Without microRNA, there will only be weak and minimal signals. But here's the challenge. Disease often involved with multiple microRNA markers. For instance, atherosclerosis is associated with multiple microRNA, including microRNA 146A, microRNA 126, and etc. If we were to detect all of these microRNA using traditional dual junction system, then that means that we need to design our new probes one by one, which is slow, resource heavy, and impractical. Therefore, we choose to incorporate machine learning into our project for the major two purposes. One is to enhance probe design efficiency. By analyzing features such as delta G, probe length, and split G4 composition, it allows our model to learn about the relationship between probe design and signals. This then enables us to design effective probe and split modes for new targets quickly, speeding up assay development. The second purpose is to extend our system to other diseases. By training on our in-house database of probes, which is microRNA interaction, this allows us to construct a generalizable prediction model. This will then, as I mentioned before, allow us to design new probes for new target way faster than before. All of these add together allow us to construct a framework that supports flexible detection across many biomarkers, which enables rapid adaptation of disease for future application.
So now I'll walk you through how our product works. We'll be providing our customers with our prepared test solution, a liquid buffer, and a screen-printed carbon electrode strip. The testing solution is the detection reagent, which we've prepared beforehand for detecting the presence of the target microRNAs. The flexible nature of the SPC strip allows it to be able to pair with a variety of electrochemical readers that are already available on the market, with various different styles and types, including a full-size version with a built-in touchscreen and a thumb-size model, consisting of a remoter and a connector. This flexibility allows the customers themselves to be able to choose the designs they prefer, and it's also great for use in clinics and health centers. Using this detection kit is simple. You first clean the bare SPC strip with water, and then rinse it with liquid buffer. Afterwards, you add the buffer again, followed by the test solution provided. Then, you plug the SPC strip into the connector, which would transmit electric signal up to a maximum distance of 20 meters to the remoter that's attached to your personal computer. The device then reads the signal, showing numerical data, allowing you to see if it's unusually high or normal. So we aim to provide our kit to health checkup centers, where it can serve as an additional diagnostic method for cardiovascular diseases during routine medical examinations, expanding this area that's often limited because they don't screen for early signs of heart disease at the molecular level. This process comes in two stages. The first stage of distribution is through the government, specifically the Ministry of Health and Welfare. By supplying our kits to the government, they can then allocate it to major central hospitals, allowing our product to be able to integrate into established healthcare systems. The second stage is where we will extend our kit to rural urban units, so people in underserved areas can also benefit from early screening. Because our kit is portable, convenient, and low cost, it can be quickly distributed and used in community health stations, helping bridge the gap between urban centers and rural care. In addition to our detection kit, we'll also provide an AI machine learning software that will help scientists also studying microRNA to accelerate biosensing assay development. We do this by reducing the time needed to collect data manually and bring convenience.
Outside of lab work, understanding the demands of those who are impacted most by cardiovascular diseases, or CVD, should be heard and addressed is a key cornerstone of our project. But in order to make sure that we're engaging with the correct audience, we must first identify and analyze several key target audiences through different aspects such as their urgency in the issue. For example, we identify mobile care stations with social worker as a priority since we can build trust and foster strong communications with the elderly populations. Additionally, we've also visited elderly care communities and hospitals at Zhanghua to get clearer insights on the issues at hand. We also interviewed local hospital and welfare staffs on rural health distribution issues and treatments. But our consultation doesn't just end there. We've also interviewed doctors, entrepreneurs, and social workers to get key takeaways such as marketing strategies and the issues in the status quo from a professional perspective throughout our human practice cycle. But we realized that these experts are not just interconnected within, but also with other experts, cooperators, and even factors such as regulatory barriers. Therefore, we utilize an actor network theory, which is a framework to comprehensively understand the relationship between different elements and how each one of them influence one another. But in order to make sure that our design reflects upon society's expectations, we've designed our procedures with the most wanted features analyzed earlier, and then experimented and concluded results on the biosensor's performances across different microRNAs. After experimentation, we've developed a 5-year Gantt chart of what we need to do in different stages, such as patent filing, clinical trials, and pilot program with hospitals, and etc. But as we execute our Gantt chart, we never stop to record experts' opinion through podcasts, present in front of companies, add to our business proposals, and even establish our very own fundraising platforms for commercialization. As for outreach, we hosted our very own experimental camp for high schoolers to get hands-on experiences at National Taiwan University. We've also participated in Taiwan's own iGEM, TGEM, with other iGEM teams from Taiwan, potential investors, and even experts. Finally, we boosted our social media presence with not only the events listed earlier, but also by promoting educational content on CVDs and microRNAs. And to improve scientific literacy, we've also taught students at Yongfu Elementary School on nucleic acids, CVDs, and biosensors' role in combating the problem so everyone can be aware of the problems and solutions at hand.
Cardiovascular disease is not just a global health crisis. It's also a deeply personal issue that continues to affect millions of lives each year. And as our lifestyles evolve and populations age, it is especially critical that we find a way for early, accessible, and accurate detection to challenge this issue. As of now, we have already collaborated with multiple biotechnology centers and clinical professionals in order to refine our project. Moving forward, we hope to be able to gain approval from Taiwan's Food and Drugs Administration in order to be able to continue moving our impact throughout the real world. After that, we hope to be able to introduce our products to not only the major central hospitals, but also ones in rural communities and indigenous healthcare systems to ensure that our product is accessible to everyone. Ultimately, we envision that our system will be able to be a part of health monitoring in both clinical and home settings. Moving forward, we hope to integrate this product into real life so that people can continue having accessible healthcare in an increasingly data-driven world. We sincerely thank you for your time and consideration, and we look forward to advancing global healthcare through our innovation in synthetic biology.